Hello friends, this is Chetna. Today we are going to learn about cloning. So basically cloning is certainly an event of 20th century. A DNA contains all the genetic information in coded chemical form to make and maintain a living cell. These things we know very well. So it can also reproduce itself given the right environment. Agree? This discovery by Watson and Crick in 1953 was taken a step further in 1997 by Dr. Ian Wilmot, the Scottish researcher, and his colleagues in creating Dolly, a lamb, through cloning. They placed the cells from the udder of a pregnant sheep, a fin dorsal eve. The name of that sheep is a fin dorsal eve. In a solution that made it difficult for them means cells, to go on dividing. So the cells were thus made passive through alive. Okay. The cells were thus made passive though alive. Separately, they took an unfertilized egg from a sheep of the same species. Which species? A fin dorsal eve, and removed its nucleus together with its DNA, but retaining intact all other chemical compounds in it. So this is the procedure they have done in 1997. Scientist or Scottish researcher Dr. Ian Wilmot. The next step was to place together both the udder cells and DNA deprived eggs and give them a mild electric shock which brought them in intimate contact. One more shock fused them together and the process of cell division began. The result was development of an embryo, a fertilized egg, and this embryo was then implanted into a womb of another Eve, same species, but the another sheep, that is a female sheep, fin dorsal Eve, female sheep of the same species, and in due course of time, Dolly was born. So this was the procedure done by Dr. Ian Wilmot, a Scottish researcher, in 1997. In the usual embryo transplants, which is now common in humans, we all know. In the usual embryo transplants, it contains, that is, embryo contains DNAs from both a male and a female, leading to a new set of DNA codes. And this embryo created by Dr. Wilmot had only one set of DNA code. So from original sheep from whose udder cells were taken, and therefore Dolly became an exact replica of that sheep. In all aspects, Dolly was thus cloned. We can say that. Now, cloning is a great biological feat which someday might allow scientists to grow new bone marrow for leukemia. Leukemia is the blood cancer leukemia patient, fight thalassemia major and other genetic diseases, or even to create healthy organs for transplanting or clone an entire individual. It is an area of tremendous interest world over, ethical reservations apart. Dolly's creation from a single reprogrammed cell has given a boost to the researchers to try different methods to establish control over genes. Thank you for watching.